We developed software for mass spectral interpretation. It's called SPec package. It mimics basically the methodology that a mass spectrometrist would use in interpreting mass spectra. It predicts the spectrum based on the structures. So it's structure-based. It basically, and rearrangements are also possible in this process. So, and the quality of these spectra are scored, so one can compare the quality of the different spectra that might be involved, the different structures that might be involved in interpreting a spectra that might have been proposed. A single spectra or a single structure or multiple structures can be inlet with a batch inlet system. A mixture of structures can be interpreted simultaneously. Structures can be enumerated, namely the substructures can be moved around in the structure, and those structures can then be interpreted against that of the spectrum. The applications of the software is clearly in any field of mass spectrometry, of small molecules, natural products, pharmaceuticals, metabolites, polysaccharides, peptides, metabolomics, and proteomics. I'll just like to give a few fast illustrations of this. This is a metabolite problem. All the arrows in red indicate where the modification of the ions, the modified ions that contain that were oxidized, are indicated, and all the blue arrows indicate regions of the spectrum where there were no modifications. So, this slide illustrates where you can draw 8 structures for a given metabolite. You can input those 8 structures into the program, and the highest scoring structure would be presumably the correct answer. In this case, that was the case. In metabolomics, a variety of natural products were proposed for a given mass spec that was obtained under metabolite conditions. And in this case, one puts in all the structures for the given natural products, and they're compared against that of the spectra. The highest scoring one turns out to be the correct one. So this is a methodology that inputs a structure, interprets the structure versus that of the spectrum. This is an illustration, a simple illustration of correlating a polysaccharide, a very complex spectrum of many, many peaks, against that of the structure, and the program automatically correlates the ions with that of the structure and scores them. This is an illustration where we have a cyclic peptide. They're very difficult to interpret the spectra. The spectra generally are very, very rich. But this program can do that automatically. And in addition, it has the feature that if there are scrambling in the ions, one can input the proposed possible scrambling substructures, these 6 substructures for that of the 6-member ring cyclic peptide, and one can interpret the scrambled spectra using this methodology. And the last one is a case of a cyclic, of a natural product. It's a modified natural peptides. It has 180 or so peaks in it, and one could use the software to essentially find the precursor ions for a given substructure. So in a nutshell, this is the SPec algorithm is a very powerful software package that...
Congratulations, Marshall. You've earned yourself another hug.